All right. Welcome everyone. This is to our September 21st Journal Club. This is our lightning session, our first one. So we're happy to have a great turnout today. Before we get started, I just have a few introductory remarks. Again, my name is Heidi Lapka. I am Director of Special Projects here at IBS. And again, welcome for attending today's Journal Club. Before we start, I would like to go over just a few housekeeping items. We are recording this session, so it will be available on the IBS website within a couple of weeks. So you'll be able to replay this session pretty soon. If you experience any technical difficulties, just reach out to me via chat, and I'll try to address those throughout the session. We have allowed for time for Q&A toward the end of the third presentation. So you can ask those questions through the Q&A session portal at the bottom of your screen, and we'll get to as many questions as possible to today's program. And then lastly, just wanted to go over some final, some ground rules. So you came in muted. Please keep yourself muted until you have a question toward the end, and then we would ask that you unmute yourself at that time. Please make sure you speak slowly and clearly when you ask those questions. And like I said, we'll get to as many questions as we can. At this time, I'm going to hand the program over to Jen Lee, who is our associate professor from the Department of Biostatistics at the University of Michigan. And he also serves as the Journal Club organizer and chair. Jen?
Thank you, Heidi. First of all, welcome everyone, and thank you very much for joining us for the very first lightning session. So the idea of this lightning session is try to find a common topic of interest to the broad audience and invite several experts to share their insights in the topic. So today we are very pleased to have three excellent speakers to talk about deep learning in statistics, which may at least from the show turnout, it seems it's of quite great interest to the general audience. And we are pleased to have Dr. Tao Sun, Wei Sun, and Francesca Mandel to join us today to present their recent paper published in Biometrics. So as a short, brief introduction to each one of the speakers, Dr. Tao Sun is an associate professor in the Department of Biostatistics and Epidemiology at the Renmin University of China. His primary research interests focus on complex biodata with high-dimensional covariance. He will present his recent paper entitled Neural Network on Interval-Censored Data with Application to the Prediction of Alzheimer's Disease. And our second speaker is going to be Dr. Wei Sun, who is a professor in the Biostatistics program at Fred Hutchinson Cancer Center. His primary research interests lie in the development of statistical and computational methods and software packages to analyze data for different types of omics studies. He will present his paper, Prioritizing Candidate Peptides for Cancer Vaccines through Predicting Peptide Presentation by HLA-I Proteins. And last but not least, Dr. Francesca Mandel is a researcher at FDA, recently graduated from the Department of Biostatistics, Epidemiology, and Informatics at the University of Pennsylvania. She will present her research on neural networks for clustered and longitudinal data using mixed effect models. So as you can see, we have a very nice lineup of speakers talking about all different aspects of this deep learning in statistics. So without further ado, I would like to invite Dr. Tao Sun to share his slides. And Tao, please go ahead. The floor is yours.
Enlarge. Okay, can I start?
Yes, please go ahead.
Thank Dr. Riddhi for the kind invitation and introduction, and thank the International Biometric Society for hosting the event. My name is Tao Sun from School of Statistics, Renmin University of China. My topic today is neural network or interval-censored data with application to the prediction of Alzheimer's disease. This is the joint work with my PhD advisor, Dr. Ying Ding from the University of Pittsburgh. I'm also very glad to see her in this journal club as well. This work is motivated by Alzheimer's disease, short for AD. AD is a neurodegenerative disorder with few effective treatments, and it's a leading cause of dementia in the elderly. AD is a progressive disorder with multiple stages, including cognitive normal, mild cognitive impairment, and eventually the last stage, AD. AD is also known as a polygenic disorder. The estimated heritability of AD is up to 79%. So it is desirable to make early prediction for AD using genetic data. So our goal is to build an accurate model to predict the time to develop AD using the risk SNPs. Our data come from the Alzheimer's disease neuroimaging initiative, namely ADNI. ADNI is a longitudinal study for AD since 2004. It investigates the clinical, imaging, and genetic biomarkers for AD. It has recruited and followed over 2,300 individuals who entered the study with CN, MCI, or AD. So for our analysis, we used 1,740 Caucasian individuals who have the genetic data. The event of interest is time to AD. We set the start time at age 55 because the late onset AD rarely happens before age 55, and also ADNI recruited the individuals after age 55. When we are analyzing the ADNI data, we realize two major attributes in this data set. First, the time to AD is subject to general interval censoring. For example, we have interval censoring if one subject progressed to AD during the study. As shown in the first line of the figure, this subject was free of AD at year 1.9, but was diagnosed to have AD at year 3.2. But we don't know the exact event time of AD. All we know is that the event happened within the interval of 1.9 and 3.2, which leads to interval-censored time to AD data. We also have right censoring when subjects are still free of AD at the last assessment time. In fact, we can regard right censoring as a special case of interval censoring because the event time lies between the last assessment time and infinity. And lastly, we also have left-censored observations for those subjects who are enrolled with AD. And in this case, the event time lies between time zero and between time zero and the enrollment time. So it's also a special case of interval censoring. So in the end, all the individuals in the ADNI data are interval censored for AD, for the time to AD. Besides interval censoring, we also have high-dimensional GWAS data. Specifically, each individual have 7.7 million SNPs. So next, I will present a neural network method that accounts for both interval censoring and the high-dimensional genetic data. Let's first define some notations. We denote T as the true event time with survival function S, and Z is the vector of covariates. For interval censored data, we observe DI for subject I that includes LI, RI, and ZI. L and R are the left and right boundaries of the observed interval. The key to our neural network method is the loss function. Here we use the negative log likelihood function plus an L1 penalty term. The penalty term helps to alleviate the overfitting issue. In the likelihood function, we have lambda L and lambda R, which are the baseline cumulative hazard function evaluated at time L and time R. Because lambda is a positive, non-decreasing, and infinite dimensional parameter, it poses great statistical challenge in its estimation, especially under the neural network framework. Besides lambda, we also have G. G is the prognostic index. we assume that G is an unknown and a non-linear function of covariance Z. So in the end, our goal is to estimate lambda L, lambda R, and G together. And also please note that this neural network is more complicated than the usual partial likelihood neural network, because the partial likelihood under right censoring only involves the parameter G, but doesn't have lambda. So the estimation lambda is the major difficulty here. To solve this problem, we propose a multiple neural network-based estimation approach. For the estimation of G, we use a feed-forward neural network, as shown in the figure below. We input the vector of covariance Z into the input layer, push it forward through the hidden layers, and then output G in the output layer. The parameter theta represents all the weights in this neural network. For the estimation of lambda L and lambda R, we propose a new type of neural network constructed by Bernstein polynomials. We denote it as BPNet. It is shown in this formula below. Specifically, BPNet takes time t as the input node. It only has one hidden layer with n+1 number of nodes. For each hidden node, we use Bernstein basis function as the hidden node activation function. And lastly, BPNet outputs the weighted sum of the hidden nodes, which is the approximation of lambda. In this BPNet, the parameter set is the collection of W, in which are the weights for the hidden nodes. So by constructing the BPNet, we are able to approximate lambda L and lambda R. Finally, once we obtain lambda hat and G hat, we are able to calculate the predicted survival probabilities for a given subject Zi at any time t. We further evaluate the prediction performance using several metrics under the interval censoring case. For example, one is the integrated Brier score, short for IBS. It is essentially a prediction error, so the smaller the better. Another is the proportion of the predicted median survival time lying outside the observed interval. We denote it as P out, also the smaller the better. Another is the absolute distance of the predicted median survival time below L or above R when the predicted median time falls outside the interval. We denote it as D out, also the smaller the better. For real application, we apply our method to the ADNI data. We first randomly split ADNI into data 1 and data 2 by the ratio of 3:1, as shown in this table below. You will use data 1 for GWAS, training, and internal validation, and use data 2 for external validation. The GWAS procedure helps us to identify top risk SNPs associated with AD progression. This slide shows the five-fold internal cross-validation results using the data 1. Each column represents one prediction model. The three panels from top to the bottom represent values from IBS, P out, and D out. The values are the smaller the better. In each panel, the scenarios from 1 to 4 indicates the increase in the number of the SNPs we included in the prediction model. As shown in this table, the last column is our proposed method denoted as NNIC. It generally performs better than the other prediction models. We observe similar findings in the external validation in data 2. We also try to interpret the model prediction result. Specifically, we calculate the individualized predictor importance values by the local interpretable model agnostic explanation method. As shown in the two figures below, they demonstrate the top 10 most important SNPs in the presence of ApoE or without ApoE. So obviously, ApoE is the most important SNP, and it plays a harmful effect on almost all the individuals. For the rest of the SNPs, we observe obvious heterogeneity within the population. For example, we just take a look at maybe the third SNP from the panel B. It is important for some, but not for all of the individuals. So, inspired by the heterogeneity within the population, we further successfully identify low and high risk subgroups by using the NNIC method. Specifically, we performed Gaussian mixture modeling on the predicted prognostic index GHAT and detected two subgroups. The two subgroups demonstrate distinct progression profiles and it is shown in the left side of the figure. On the other hand, we could not detect subgroups using the random forest method under interval-censored. So in the end, we hope that our prediction model, together with the interpretation and the subgroup identification, will be helpful for early prevention and clinical management of the AD patients. So in conclusion, our work is the first study of AD prediction that appropriately handles interval censoring and fully utilizes the wealthy GWAS predictors in the complete AD data. Specifically, the model predicts AD risks from an early age, as early as 55, and improves the quality of life before and during disease progression. It also achieves higher prediction accuracy in this specific application than several existing methods, and also identifies high and low risk subgroups. That's all for my presentation. Thank you for your attention.
Thank you, Tao. Nice presentation. So let's move on to our next speaker, Dr. Wei Sun. I'm going to talk about how to use deep learning in cancer vaccine studies. So Wei, please go ahead.
Sorry, I think since I re-logged in, you probably need to give me the permission again to
sure.
share screen.
So let me make you a co-host. Yep. It's now.
Okay. All right. Can everyone see my screen? Okay. All right. Great. So thanks again for the invitation, and I guess organized this very interesting lighting session. So topic of my talk will be prioritizing candidate peptides for cancer vaccine. Through the predicting peptide presentation by HLA-I proteins. So the two pictures here, I'm at Fred Hutch at Seattle. The two pictures I saw here is on the upper right corner is a Fred Hutch campus, and then on the bottom is University of Washington. So I'm also affiliated faculty at University of Washington and the University of North Carolina. Okay, so some background. Neoantigen. This is maybe a new topic for many of you. Feel free to ask questions in chat, or I'm okay also ask in just during the talk. So here is to illustrate a tumor cell. And then in tumor cell, the very important factor make tumor cell different other cells is the somatic mutation. So there's a mutation in the DNA sequence indicated by the red bar. And then this mutation become part of a protein. And then this protein, like all the proteins in cell, after a certain time, they are going to be break up into small peptides. So small fragments of protein amino acid sequence. And then here the color red again indicate the peptides which have the somatic mutation, because the somatic mutation change the amino acid in the peptides. And then this peptides is going to be transported to cell surface by this molecule called MHC class 1. It's a major histocompatibility complex. For human, also known as human leukocyte antigens. And then at cell surface, the MHC or HLA present the peptides to be recognized by the immune system. So this mutated peptides is what we call neoantigen, because that is the factor that making the immune cell is able to recognize tumor cell. And then the part of immune cell recognize tumor cell is super important because like every day we have 1 to 10, 5 to 10 somatic mutations in our body. And then the reason they did not become cancer early on is because the immune system survey the whole human body and find those somatic mutations and then kill those cells. So some background on cancer vaccine. The cancer vaccine is not like the vaccine for, say, a virus infection. The cancer vaccine here is for treatment. It's a therapeutic vaccine. So what it does is personalize. You got one patient, you take the tumor sample, blood sample, and then do DNA, RNA sequencing, identify the tumor-specific mutations, and then do HLA typing. You know what are the HLA alleles of this individual, and this is the key part to predict personalized HLA binding peptides. And this is basically the focus of our work. So once you predict those peptides, then you can use those peptides as vaccine. You don't need to get perfect prediction because the vaccine usually including like 10 or 20 such mutated peptides. So as long as one of them will induce immune response, and then this will help the immune system better recognize tumor. So this is a very active research area, and I'm very optimistic this will be part of the immunotherapy in the near future. And now let's back to the problem of predicting which new antigen presented in the cell surface by HLA. There are two types of HLAs, class 1 and class 2. And then each cell present up to 6 HLA-1 proteins. Because this protein can be encoded by 3 genes, HLA, B, C, 3 genes. And those HLA genes are highly polymorphic. It means there are many different copies in the human genome. And then use the recently developed mass spectrum data, we can identify those peptides. They tend to be presented by HLA, and then use those data to train a neural network. The challenge is those mass spectrum data, what they give you is a mixture of peptides. They can be presented by any one of the HLA allele of individual. So let me go through a little bit more detail about the HLA. Again, some of you may be familiar with HLA, but just make sure we're on the same page. So HLA protein can be encoded by one or 3 genes, and the HLA-2 proteins are encoded by 1 or 3 gene pairs. In this work, we actually focus on HLA-1 protein. We have another work focus on HLA-2 protein. So in chromosome 6, this HLA region, it's a small region. It's really the most polymorphic region in the human genome. And if you look at a family pedigree, then you can see this is the number 1 here. This one individual inherited the HLA-1, 2 from the parents. So you can see for HLA-A, B, C, there are like 3 genes because they're maternal and paternal copies, so can have up to 6 different HLA genes. And except this one DRA in HLA-2, all the HLA-1 and 2 genes are very polymorphic. There are like more than 10,000 different versions of them. So this is one way to generate a diversity in the human genome in terms of our immune response. Think about that. There's no virus can kill us all because we have so different HLA alleles. Okay, now let's back to the work here about predicting the interaction between the peptide and the HLA. So the input of the neural network are the peptide sequence. So basically each letter here is 1 amino acid. There are 20 possible choice. So you got the peptide sequence. And then for the HLA, they are longer proteins. We only take 34 positions. They interact with the peptide. For example, this is the amino acid sequence. And now this is one reason we want to use neural network, because the input data is like non-standard. They are amino acid sequence. We can encode them numerically to this one hot encoding. Basically, if there are 15 amino acid, because there are like 20 possible choice for each amino acid. So for one position, we encode is like a vector of length 21. So that's called one hot encoding. For one position, indicate one amino acid. So for example, if amino acid F here, then for the position F is 1, all the other position is 0. So we can put this sequence to this is the data matrix. And then the HLA pseudo sequence is another data matrix. And then we just put them together as one big vector, and then go through a neural network. It's not a deep neural network, just a couple layers. And then we consider a few different neural network architectures with a single layer with a 200, 400, or 800 nodes. We have also tried a neural network with 2 or more layers. They don't really help much. And then we try to do this ensemble prediction. We combine 15 neural networks, and for each the architecture, we try to train this using 1 of 5 splits of the training data. So this is well known that any neural network you have, if you do ensemble, you can always improve your prediction by like a few percentage. And then at the end, we got a score predict whether the peptide is presented by a particular HLA allele. An important advantage I should mention here is we use the HLA sequence to represent the HLA, so that make our result easy generated to those rare HLA alleles, as long as we know their sequence. Okay, so a little bit more detail on the statistical model. Now, assume we got the binding peptides on N individuals, and then for one sample, for one individual, we have say Ki HLA alleles, and then Ji peptides, and let Xij and theta Ik denote their corresponding sequence. So the Zi is the number of peptides. It can be from tens to thousands. Ki is the number of HLA-I alleles. So they vary from 1 to 6. Li, we select Li non-binders. So basically, we randomly sample peptides from human protein, and then we randomly assign them to one HLA. And then we use this neural network to quantify the likelihood that this peptide Xij is presented by the HLA with the sequence theta Ik. And then we can write out the likelihood function. It's really standard mixture distribution likelihood in the first part. So you ignore the first product, it's just the product across N individuals. Then within individual, you look at all the peptides. So that's a product. And then for each peptide, you know, this is a mixture distribution because we do not know which HLA-I allele it's presented by. And then for the non-binder, we just use 1 minus F as the density. And then we just apply a standard EM algorithm to fit that. And the only challenge here is in the M step, how do you estimate this F? So that's part we use the neural network. The objective function, the Q function in the M step is exactly cross entropy loss for a neural network. Okay, now let me talk a little bit more on the data. So we train our model using the data, both single allele and multi-allele. Single allele data means for the peptides, we know which HLA allele it's associated with. The multi-allele means we do not know. So there are 130, we know the single allele data. We also use the single allele data to evaluate. The figure C here is the, for 2 HLA allele, we check whether they have, they present similar peptides. And then we use that to define a distance to do hierarchical clustering. And then we find after the hierarchical clustering, the HLA allele, they have similar name, will be clustered together. That means if they have similar name, they present similar peptides. So this is an illustration that, I mean, the HLA name is closely related with their sequence. So this is an illustration that the peptides presented by the HLA allele are closely related with their sequence. So by the way, this is the cover picture, I think, for this issue of biometrics. and then also the multi-allele data. So for each sample, we could have 3 to 6 HLA alleles. Okay, and then this is the result when we compare our method with one very popular method, that MHC PEN 4.1. So this is in the independent set-aside testing data. So you can see that our method can achieve higher AUC. And then this is the data. In the testing data, there are multiple samples. So this in the second panel B, we compare the performance of our method we call peppermint versus net MHC PEN for each sample. Here, I think each cell line separately. So we can see for most cell lines, we can have better performance, probably except one. And then this panel C is comparing another method called the MHC flurry. It's also a popular method using neural network, but it's trained using the single allele data, not multi-allele data. So you can see we do perform better than MHC flurry in most of the samples. Okay, and then we look at one real data in melanoma cancer patients, and then check how can we predict or select the somatic mutation for candidate cancer vaccine. So this is a little bit messy here. We look at the panel A here. Each point is one peptide, and then the X and the Y axis are the prediction score, like how likely they're going to be presented by at least one of the HLA allele of that individual. And then panel B is the same thing for peppermint. So what we want to find are the points in the upper left corner. Basically, the peptide have a high prediction score by peppermint, but low prediction score by the reference sequence without mutation. And then we also evaluate called the new antigen burden. Basically, if we predict how many new antigen each person has, and that's a burden, and then we check its association with this cytolytic score. Basically, it's derived from gene expression indicating how active is the immune system. So we compare our three methods, the mutation burden, new antigen burden by net MHC, and the new antigen burden by peppermint. So we do see the peppermint give a better result. The difference is not huge. I wouldn't say too much about that, but we do see a sign that we can perform better. And then the panel C is just saying if you try to draw the threshold to select new antigen burden with a different threshold, then consistently we see our method can achieve smaller p-value than net MHC PEN. With that, I will acknowledge this work is led by Laura Zhou. This is part of her dissertation. She is currently assistant professor of biostatistics and health data science at Indiana University School of Medicine. It's also a collaboration with Dr. Fei Zou, professor of biostatistics in UNC Chapel Hill. So thank you for your attention.
Thank you, Wei. Nice presentation. Our next speaker is going to be Dr. Francesca Mandel. So Francesca, feel free to share a screen and go ahead. Wei, if you could please unshare. Yep.
I think you need to make me a host again. It's saying it's disabled for me.
Do that.
There we go.
Yep.
Okay. So hi, everyone. I'm Francesca. Today, I'm talking about neural nets for clustered and longitudinal data. And this is joint work with Dr. Ying Barnett, who's also here with us today. So this work was motivated by developments in the world of mobile health or mHealth data. And this is data that's collected from mobile devices. So this, for example, could be smartphones, which for many of us is something that we carry with us everywhere we go, all the time, every day. And so this creates this really unique opportunity to collect data in more naturalistic rather than clinical settings. And this is really helpful in particular for characterizing different mental health conditions. So what this graphic here is representing is that our phones can collect a wide variety of data on us. So for example, we can collect GPS data and we can use that to track a subject's location throughout the day. We can use accelerometer data to track physical activity levels or sleep patterns. And we can even pull information from call and text logs if we want to measure the level of social interaction that a subject has in a given day. And so you can kind of see how these types of features could relate to mental health conditions. And all of this data that's collected by our phones is collected passively. So it's not requiring us as individuals to actively provide this information. It's just being collected in the background. And this is great for two reasons. One, it just reduces the burden on the patient, which is always a great thing. But secondarily, it also reduces the bias that we know exists in a lot of patient-reported outcomes. So, if we were trying to characterize sleep, unlike maybe a traditional sleep study where a subject would come in, maybe weekly and have to report back on 7 days of sleep, here this is just being collected by the phone in the moment. And so it's a more objective way of capturing this information. And so the goal with mobile health is that we want to translate all of this data into biomedical and clinical insights. And so in that vein, this term digital phenotyping has been coined, and it's defined as the moment-by-moment quantification of the individual level human phenotype in situ using data from personal digital devices. And I'll note here that it's called digital phenotyping rather than digital phenotype because we think of this as something that is changing over time. It's not static. So we're not going to define it once and have it follow a patient throughout the study. It's something that could be changing, potentially daily. So one of the areas where mHealth data is particularly useful is in psychiatry. And I talked about this a little on the previous slide, but smartphone data is really ideal for capturing various social and behavioral dimensions of psychiatric diseases. So a lot of psychiatric disorders, they're characterized by a chronic course, and then there's a very high risk of recurrence. And so what this means is that care typically consists of waiting for some type of major crisis to occur, which generally leads to hospitalization. After that, patients might be discharged to outpatient care if it's available, but ultimately there's very high readmission rates. And so the reason that care usually takes this form is because it's actually very difficult to determine in advance when a relapse might happen. But the idea here is that new screening efforts using digital phenotyping could potentially enable earlier detection and allow us to deliver some type of intervention before the relapse occurs. So just to kind of illustrate this in practice, suppose we had a single feature pulled from smartphone data on a single individual. So this plot here on the bottom would be over the course of 3 weeks, what this feature looks like for one subject. So if this was something like time spent at home, you could kind of see this clear weekly pattern emerge, where maybe on the 2 weekend days, they spend a lot of time at home. And for the 5 weekdays, they spend less time at home. Maybe they go into an office. And so if we could model this weekly pattern really well, and that's the purpose of today's talk, is what's the right method to do that. But if we can do that well, we can also detect changes from that pattern. So for example, on day 19 here, something different seems to be happening. And if we actually look at the deviation from trend, you can see it's very clear here. And so this might be indicative of some type of change in their health status. And if that's the case, we'd want to do something about that because we know in advance. So really, this is all to say that the larger goal here is just to use these data to better monitor health status and give better care. So specifically for this work that I'm talking about today, we were motivated by this small mobile health pilot study on patients with schizophrenia. So in this study, the subjects installed an app on their phone, and then we followed them daily for a few months. So this graphic here is representing the data that we would have on a single individual. So the columns here are days. You can see we have several weeks of observation. And then the rows are features. And we can think of the data in the study as coming from 2 different data streams. So the first would be the passively collected data. And this is information that's just collected in the background on the phone. The patient is not actively providing any information for us. And so here, this is pulled from call and text logs, as well as GPS and accelerometer data. And so you can see these features are represented in the green, the blue, and the pink. And so we have these daily summary measures of features such as time spent on the phone, or distance traveled from their house. And then the other form of data is the actively collected data, and that's represented by the orange here at the bottom. These were survey questions that we set as push notifications that ask the subjects to report on their sleep or their anxiety or their mood. And so the goal here is we wanted to develop a model that could use all of the passively collected data to predict the actively collected data. So specifically what we're going to do is use the GPS and accelerometer data to predict future depression and anxiety levels. And the idea is that if we were to use this in some type of ongoing clinical practice, we'd want to collect the active data for a few months to establish a baseline. And then on an ongoing basis, we'd use the passively collected data to predict those active data. And this way, this subject wouldn't have to provide anything for us. And if we can do a good job at predicting those things like depression and anxiety changes, we can really monitor their condition. So the question becomes how best to model these data. It's longitudinally collected. We have very data-rich participants, so we want to be using that information as best we can. So to reiterate, the goal here is to predict future depression and anxiety levels using all of the passively collected smartphone data. And in order to do that, we kind of have three key considerations. The first is that we want accurate prediction. We want to be able to detect very small changes in depression and anxiety. And so one of the ways that we could improve prediction is to leverage individual information alongside study level effects. Secondarily, this is longitudinal data, so it's collected at different intervals and there's different lengths of follow-up for each subject. So we need a method that's flexible to handle that. And then last, and perhaps most important, is that we have a belief that there's going to be these complex associations between our clinical outcomes and these smartphone data. I think it's very reasonable for people to understand that a feature like time spent at home could definitely be correlated with things like anxiety and depression. But exactly how those things are related is hard to say in advance. It's probably not something simple and linear. And so, we know that mental health is complex and it's varied. So we really want like a data-driven approach that could flexibly model what might be highly nonlinear associations. So with those considerations in mind, if we think about the methods that are available to us, if we were focused on accurate prediction and allowing for complexity, neural nets would be an obvious choice here. We know that they are really powerful predictive models. But there is a caveat, which is that they assume the data are independent. And since we have repeated measures, this is going to be a clearly violated assumption. There are, of course, architectures that have been proposed for data collected over time, for example, recurrent neural nets, and we talk about those in the paper, but ultimately none are really working well, specifically for the longitudinal setting. On the other side of things, if we were just thinking about the fact that we have correlated data, an obvious choice here would be traditional generalized linear mixed models, GLMMs. They can handle various types of correlated data, but the limitation here is that they are limited to linear fixed effects. So we're not going to be able to capture that complexity that we expect. And so really just the goal with this project here is to combine the two. So combine neural nets with GLMMs, kind of pull the best of both worlds. And this way we can be simultaneously accounting for correlation and complexity and get really good predictions. So what we propose in this paper is this generalized neural network mixed model, GNMM for short. And what we're doing here is just incorporating that random effect structure from GLMMs into a neural net. And this allows us to better predict this nonlinear correlated data. So how we actually do this at a high level, we start with the GLMM. So this is a formula very familiar to all of us. We have a linear fixed effect x beta, this random effect zb linked to this outcome mu through the link function g. And what we're doing is just replacing that linear fixed effect with a nonlinear function. And we're going to define that function as a feed-forward neural net. So this function f, it's a function of our data x, as well as the network parameters, the weights, and the biases. And so it's just this swap here from something linear to something nonlinear, and specifically it's a neural net. And then we can write this more as the way we typically see with neural networks, where we have this output is a nonlinear function of the final layer of the network. And here we still have that random effect. So it's omega alpha plus delta from the network. We add our random effect, and that all gets put into this activation function G naught, which you can think of here. This is like the inverse link function from the GLMM framework. And I won't get into the math just of how we actually train this model, but I will point out that it is a joint estimation procedure. So we are simultaneously estimating the neural net parameters and the random effect. Just to give a visual of what this model looks like. So in black on the left, this is just a traditional feed-forward neural net. It can be any size, any depth. And where we're making the adjustment is just in red. It's at the last layer here, where we add in the random effect before we take that final nonlinear activation function. So that's how we set up the model. And in the paper, we have simulations that look at this model compared to regular feed-forward neural nets, as well as to traditional mixed models. And we see that generally when we have nonlinear correlated data, our method is much higher accuracy. And even in the simpler case where the data is either uncorrelated or linear, this is still a method that can work well. But I think what's most interesting to talk about is how the model performs on the data that actually motivated it. So we were trying to predict symptoms in schizophrenic patients. So we had this, as I mentioned, this pilot study with 15 subjects with a diagnosis of schizophrenia. And we for a 90-day period. And interesting to note is that we have an average of 53 days of observation per subject. So this is actually quite a bit of subject level information here to use. And then we're using 15 daily mobility measures that are collected from passive GPS and accelerometer data. So these are going to be features like distance traveled in a day or time spent at home. And then we are predicting depression and anxiety levels at the final day of follow-up. So we're using a subject's entire history up until the final day to predict what their depression or anxiety level would be at that final day. And that's on a scale of 0 to 3. And so we're comparing our proposed model here, the GNMM model, to traditional mixed models, GLMM, as well as to just a regular feed-forward neural net that's not accounting for correlation. And I'll mention here is that we have 2 different size GNMM models, 1 and 2 here. These are with 1 and 2 hidden layers. And I think that might seem strange at first because we're talking about machine learning. So you typically think of deep learning, these very highly parameterized models. And that's all really useful to do something very complicated like image classification. But here we're thinking about this specifically for the setting of longitudinal data. So in addition to probably just not having as much data as we would in some of those other machine learning tasks, so we might not be able to stably estimate such a highly parameterized model, we also just don't need that level of complexity. We're looking at this kind of in the setting of maybe just one step in complexity above a linear model. And so if we have just 1 or 2 layers, this is generally sufficient to model our data. And it's also just better scaled to how much data we actually have to estimate these parameters. So here we're comparing these 2 GNMM models, as I said, to the GLMM and ANN. And we can see just on the surface is that GNMM has the lowest prediction error here, which is to be expected. This is correlated data with these complex associations. And so this is the model that's capturing both of those things. But if we look at some pairwise comparisons, I think it builds a little intuition behind why this works well. So if you compare just GNMM to GLMM, there is a small reduction here in prediction error, which is we can attribute solely to whether we account for nonlinearity. So clearly there's a gain here in going from a linear model to a nonlinear model. But more interesting is if you compare GNMM to just a traditional neural net. And here there's this much larger reduction in prediction error, which is attributed solely to whether we account for correlation. And so we can see clearly here that there is very strong individual level effects. And if we account for that, we're going to vastly improve our prediction. And for some context, as I said, depression and anxiety are scored 0 to 3. So having this prediction error of greater than 1 for the traditional neural net is a pretty meaningful difference, actually. And I think this kind of goes against that notion of, well, you could throw anything into a neural net and get good prediction. It still matters what the assumptions of our model are. And if we are violating those, our prediction is definitely going to suffer from it. And so if we kind of think deeply about what these models are doing, the ANN model is relying solely on population level effects to predict these individual health outcomes. But the GNMM model is actually modeling those individual trajectories, but still leveraging information from the study population. And so really what we see is that GNM is really useful here because it's pulling these elements both from more traditional statistical models as well as from machine learning. So just to wrap up here, at GNMM, we're essentially extending the traditional neural net framework to include a random effect component. And this allows us to better predict longitudinal and other types of correlated data. And I think this is really useful kind of in this new data world where we have different sources of longitudinal data like mHealth studies that have far more densely measured follow-up periods than traditional longitudinal studies. And so we have this opportunity to really model more complexity in individual patient histories. And so this work was back in 2021. As I said, this was joint work with Dr. Ian Barnett and Dr. Riddhi Pratim Ghosh. And we have code available for this on GitHub for implementing the model. So thank you for listening. Happy to answer any questions.
Thank you, Francesca. Very nice presentation. And I would like to thank all the speakers for this excellent work and the presentation. Now the floor is open. So if you have any question for any speaker, feel free to unmute yourself and speak up, or given we have a large audience, maybe you can raise your hand, use the raise hand feature. And so I would like to start by asking a general question for all the speakers. If you could share with us your take on why you choose to use neural network or in general this deep learning approach in the first place compared to other more conventional statistical methods, prediction methods, and what are the caveats that you have encountered in your procedure? So maybe we can start with Tao.
Sure. Thank you. Actually, this is the second neural network method work I did. In 2020, I applied neural network survival model to the genetic data from the AMD patients. So I realized that at that time that neural network seems to have better prediction performance than other machine learning models in the genetic data. So when it comes to interval-censored data, so it's very natural for me to think of using neural network to apply to the genetic data. That's the very first motivation for developing this method.
Great. Any caveats you say while you are doing this? How? Do you say any caveats or any?
Oh, caveats. Of course, the training process takes time, and the need to select the hyperparameters, and also it takes time to to train the model.
Got it. Thank you. Wei, do you have anything to add?
Yeah, I mean, in the problem I described, they are basically the input to amino acid sequence. I think the one motivation is there may be some nonlinear relations. There may be interaction in any two positions. So that make the whole parameter space is very large. If the effect is totally linear, if we just look at position weight matrix, look at each position, what's the likely amino acid, then we probably do not need a neural network. But because there are any possible interactions, maybe the interaction between like one amino acid to two other amino acids, there are many possibilities. So that's where the neural network could have some advantage. And this is also a kind of well-studied problem, partly because of the popular immunotherapy. Many people are interested in this question. And then traditionally, people have shown neural network actually can perform better than other methods like support vector machine, regression, other type of techniques.
Great. Francesca?
Yeah, I would definitely reiterate what Wei said, especially about interaction effects. I know for us, we have very highly correlated features that are likely interacting and how they predict the outcome. And, using neural networks allows us to not have to specify that in advance and really let the data drive that. I think on the other side is the caveat, like Tao said, it's just more complicated. There's a lot more to choose, maybe a slightly harder sell for clinicians as well to use in practice.
Yeah, thank you. So that's very good guidance, at least to someone like me who haven't really used the deep learning tools that much. So it's a good heads up on that. So I saw some question from the chat. One question I believe is for Francesca. So it's that the problem with digital data is running out of battery or leaving the phone somewhere you are not, which may be due to illness. Do you consider this to be missing at random? For the latter, you leave the phone somewhere due to illness. Is it missing at random assumption of property?
Yeah, this is a great question. I will say we haven't actually done a lot of work on how to deal with the missing data. For most of this paper, we were doing complete case, but it's definitely something we thought about. I think with the first example there, you could maybe argue that that's missing completely at random because that's some type of almost device malfunction. But certainly for the second, the idea of leaving the phone somewhere you're not, that could definitely be related to disease status. And obviously that's a hard thing to verify. So I don't necessarily have a good answer to it. But I think this issue of missing data with digital health technologies is still kind of maybe a bit of an open question. Some are easy to characterize as completely at random, but a lot of these are likely related to the outcome.
Right. And another question or comments is you may wish to look into GAMLSS. I'm not sure what that is, but maybe we can look into that, see how that's compared to what presented here. Any other questions from the audience? You can raise your hand or you can put it in the chat.
Maybe I'll just give a comment. I think we talk about the computational neural network. It is take more time to get familiar, but one thing that people may also feel uncomfortable for neural network, it seems there are many tuning parameters to tune. I think one experience from our study is most hyperparameter actually work okay. Like if you want to do classification, you'll see many tuning parameters give you AUC like 0.7 to 0.8. like, I mean, it's somehow better than the other, but it's not super sensitive because there are some latent structure in the data can be captured by the neural network. So it is tuning is a work, but I don't feel it's something like very tricky. Yeah.
It's good to know. Thank you. Any other questions, comments from the audience?
Hi, I have a question for Francesca. So yeah, that's a very nice talk. Like in the mixed model, applying the deep learning to the mixed model framework. So I have been working on like the using the joint model of longitudinal and small outcome. So I wonder if your method can be applied in that situation. For example, to do some dynamic prediction or survival outcome using longitudinal biomarkers.
It's a very interesting question. I think probably with some adaptations, but that's actually not an area I'm super familiar with. But I think that's an interesting idea. You could maybe make some extensions to that.
Thank you.
Okay, Pamela, Shaw.
Hi, I'll just say that first, it's been really interesting hour or so. Really enjoyed all the talks. I guess, Francesca, you have the benefit of being closest to the question and answer period. So I have a question for you, and maybe it relates to the chat question as well, the GAMLSS. Like what I was thinking maybe, which was along the lines of whoever typed that in the chat, if your network is relatively shallow, and I will say full disclosure, I have a very shallow understanding of deep learning, but if you have a very shallow network, then how is it different than, or what are the benefits of, it sounds like just simply adding some splines and some non-parametric bits to your mixed model, like you compared a generalized linear mixed model to this neural network, and there's something in between, right? Just like a more complex thing. Yeah, and maybe
I think it's a great question, because you could also think of using, we can extend generalized additive models to have random effects. Like there's a lot of options.
Yeah.
And I think one of the things that we kind of came up against is that with a lot of those other in-between models, there's still a level of specification of how you want to either specify interaction effects. So like in the paper, we do also consider GLMMs with 3, 5, certain numbers of interaction effects based on what we might expect among the data. But I think ultimately, as statisticians, we don't always know clinically what would be relevant to include. So we kind of see the benefit with the neural net is you don't have to specify that. Hopefully the network is going to pick up whatever type of interaction effects. But I think, again, it depends on the complexity of the data. I definitely think something like a GAM or some of these other proposed things might be the good in-between where you don't have to jump to the neural net. But I think if you're a little unclear exactly what might be the best way to model the data and what might be happening among the different features, this is kind of a flexible data-driven way to do it. But I agree with you. I think there's kind of a gradient here where there's a lot in between going from the linear model to a full-on neural net.
No, that's great. And I don't know, I had the mic here. If any of you had a recommendation for like neural nets for dummies, like on how to get started, I don't know, typing a favorite reference into the chat or something might be helpful for some of us who are maybe looking at our first applications in neural nets. But, great talks, everyone. Thanks.
Yeah, that's a great question. So if any one of you have any general suggestions of how, we can get started with this, very interesting topic, you can put in the chat so everyone can have access to. All right. Any other questions? We're right around 12 p.m. Eastern time. So I'll give last chance for questions. So you can always ask questions later on, using the functions on IBS platforms, but I think we are about time to wrap up. Again, I would like to thank all 3 speakers for their wonderful work and wonderful talks. And also want to make an announcement that our next regular journal club will take place on the 3rd Thursday of October, which is going to be October the 19th. And our next lightning session will take place on the following month, which is November the 16th. So we are looking for interesting topics and talks. So if you have any suggestions about the topics or speakers, please don't hesitate to let me know directly. Otherwise, please stay tuned. I will now hand it back over to the International Business Office, which manages the logistics of the Journal Club. So Heidi, please.
Yeah, thanks, Jen, and thanks to our presenters again for a great session. We hope you all enjoyed the lightning session. If you'd like to continue the conversation or have any additional questions, we encourage you to head on over to the community. Make sure your subject indicates Journal Club, and we'll make sure Jen and the presenters see some of those questions that come through. As I mentioned, a recording will be available in 2 weeks on the website, so check that out. Also, we want to hear from you. We want your feedback on future sessions. So please share those suggestions via the QR code listed on the screen. And we look forward to seeing you at an upcoming session. Thanks again and have a great week. You may now exit the session.
Thank you, everyone.
Thanks.
For the rest of the day. Thank you.
Thank you. Well done.
Thank you.